Okay, so today we're going to really dig into diabetic retinopathy, or DR. But,
Right.
I think everyone listening already knows just how important DR is, but we wanted to do a deep dive into some of the newest info and research on it.
Yeah, for sure. It's super relevant for everyone, not just retina specialists.
Exactly. General ophthalmologists are seeing these patients all the time, too. For this deep dive, we've gone through the latest research articles and guidelines, trying to really pull out the stuff that's going to directly impact how we manage these patients day to day.
It can get pretty overwhelming trying to stay on top of all the new research.
Oh, yeah, for sure. So our goal here is to sort of distill everything down to the most crucial takeaways for ophthalmologists and retina specialists.
That sounds great. I think everyone could use a DR refresher.
Definitely. Um, so maybe a good place to start would be just a quick recap of the basics.
Sure.
Like, why is diabetic retinopathy such a big deal?
Well, simply put, it's a leading cause of vision loss worldwide in working age adults.
Yeah, that's a huge problem.
Yeah, and it's a microvascular complication of diabetes mellitus.
Right.
And the thing is, it's not just having diabetes that puts you at risk. It's things like how long you've had diabetes, how well your blood sugar's been controlled over the years.
Uh-huh.
If you have high blood pressure on top of that, and just getting older in general, all of that increases your chances of developing DIR.
Okay, so those are the main risk factors that we always need to be thinking about.
Absolutely.
Yeah.
And it's always good to remind our patients about that too.
Definitely. Now, in terms of classifying DR, how do we usually categorize it clinically?
At the most basic level, we split it into non-proliferative diabetic retinopathy, or NPDR,
Right.
and then proliferative diabetic retinopathy, which is PDR.
Okay, so NPDR is sort of the earlier stage.
Right, exactly.
And then PDR is when things start to get really serious.
Exactly. PDR is where we start to see neovascularization.
Right, those abnormal blood vessels.
Yeah, which can lead to all sorts of complications like vitreous hemorrhage and retinal detachment.
Yeah.
But, we can't forget about diabetic macular edema, or DME.
Right, DME.
Which can happen at any stage of DIR.
So DME isn't necessarily tied to whether it's NPDR or PDR.
Exactly.
Okay, so it's kind of its own separate thing, but
Wow.
can occur alongside either NPDR or PDR.
Right.
Okay, that makes sense. Now, let's delve into the pathophysiology a bit. It sounds like chronic high blood sugar is the real culprit here, right? What exactly is going on at the cellular level when someone has chronically elevated glucose?
Well, there are actually a bunch of different pathways that get activated and go haywire because of all that extra glucose floating around.
Okay.
So one important one is the activation of these alternative glucose metabolism pathways. Like, normally glucose gets metabolized in a specific way,
Right.
but when there's too much of it, it starts spilling over into these other pathways, and those pathways can end up causing a lot of damage.
Interesting. So it's not just the glucose itself that's the problem, but what happens when the cells try to deal with all that excess glucose?
Exactly. It forces them into these alternative metabolic pathways.
Okay, so what are some of these pathways that we should be aware of?
One is the polyol pathway.
Okay.
Basically, excess glucose gets converted into sorbitol, which builds up inside the cells,
Uh-huh.
and that buildup causes osmotic stress, basically drawing water into the cells and making them swell up, which obviously isn't good.
Makes sense.
Plus, the conversion of glucose to sorbitol uses up a coenzyme called NADPH,
Uh-huh.
and that NADPH is actually really important for protecting cells from oxidative damage, so depleting it leaves them more vulnerable to those harmful free radicals.
Okay, so it's like a double whammy,
Yeah.
damaging the cells directly and also weakening their defenses.
Yeah, exactly.
So you're saying even at the early stages of diabetes, before we even see any clinical signs of retinopathy, this damage is already starting to happen at the cellular level.
Exactly.
That's really important to keep in mind.
Yeah, it highlights why tight blood sugar control is so important right from the get-go.
Definitely. So we talked about sorbitol buildup, but you also mentioned something called advanced glycation end products, or AGs.
Right, AGs.
What are those exactly, and what role do they play in DICAR?
So AGs form when glucose reacts with proteins or lipids.
Mm-hmm.
It's kind of like caramelization, but happening inside your body.
Okay.
And these AGs are bad news.
How so?
Well, for one, they can cross-link proteins. So imagine two proteins that are normally separate getting glued together by an AG.
Uh-huh.
That messes up their structure and function, and that can have all sorts of downstream effects.
So it's like they're disrupting the normal architecture of the tissues.
Exactly.
Okay.
Like in the retina, AGs can stiffen up the basement membranes of blood vessels,
Uh-huh.
making them leaky and dysfunctional.
That makes sense.
But AGs can also bind to specific receptors on cells called RAGE receptors.
Rage.
Yeah, it stands for Receptor for Advanced Glycation End Products.
Oh, okay.
And when AGs bind to these receptors, they trigger a whole cascade of bad things.
Like what?
Well, they ramp up oxidative stress, which we already know is bad.
Right.
But they also stimulate the release of all these inflammatory molecules.
So it's fueling that inflammatory fire we talked about earlier.
Exactly. And AGs are linked to the loss of pericytes, which are those cells that provide support to the capillaries in the retina.
So they're weakening the very foundation of the retinal blood vessel.
Exactly. And that can lead to capillary closure and all sorts of problems down the line.
Okay, so AGEs are a major player in DR development. What about protein kinase C activation? How does that fit into the picture?
Well, PKC, it's a family of enzymes, and they're involved in a ton of different cellular processes. But in the context of DR, the PKC beta isoform is particularly important.
Okay.
And hyperglycemia leads to the buildup of this molecule called diacylglycerol, or DAG. And DAG directly activates PKC beta.
Okay.
So you get this overactivation of PKC beta in the cells of the retina.
And what does that lead to?
Well, it kicks off this cascade of events that ends up promoting inflammation and oxidative stress.
So yet another pathway contributing to those two key processes.
Yeah, exactly.
Okay.
And one of the really important things that PKC beta does is it stimulates the production of VEGF.
VEGF, that's vascular endothelial growth factor, right?
Right.
The one that promotes the growth of new blood vessels.
Exactly. So it plays a major role in the neovascularization we see in PDR.
Okay, so PKC beta activation is like hitting the gas pedal on VEGF production.
Exactly.
And on top of that, it also ramps up the activity of this enzyme called NADPH oxidase.
Which, as we discussed earlier, contributes to oxidative stress.
Right, so another negative feedback loop.
Yeah, unfortunately.
Okay, so we've talked about the polyol pathway, AGEs,
Yeah.
and KKC activation. Are there any other important pathways we should be aware of?
Yeah, one more that's kind of under the radar, but it's important, is the hexosamine pathway.
The kesamine pathway. I'm not sure I'm familiar with that one.
It might not be as well known as the others, but it's actually a key regulator of a bunch of cellular processes.
Okay.
Think of it like a fine-tuning mechanism for the cell.
Okay.
And high glucose basically hijacks this pathway and throws everything off.
How so?
So normally, some glucose gets shunted into this pathway and converted into a molecule called UDP-GLC-NSA.
Okay.
And this molecule is important for something called OGLC-NSation.
OGLC-NSation. Wow, that's a mouthful.
Yeah, it's a bit of a tongue twister. Basically, it's a process where this sugar molecule, GLCNS, gets attached to proteins.
Okay.
Kind of like phosphorylation, but with a sugar instead of a phosphate group.
Interesting.
And this OGLCNSation can change the activity of proteins, turning them on or off, or changing how they interact with other molecules.
Okay.
So when you have high glucose, you get more of this UDP-GLCNS being produced. And that means more proteins are getting OGLCNacylated.
Oh.
And in the context of DR, this has a bunch of negative effects.
Like what?
Well, for example, it can increase the activity of certain transcription factors.
Transcription factors, those are the proteins that control gene expression, right?
Exactly. So
Okay.
one transcription factor that gets affected is SP1.
SP1.
Yeah, and when SP1 gets OGLC-acylated, it actually starts to produce more of these pro-fibrotic factors like TGF-beta and PI1.
Oh, so it's promoting fibrosis in the retina.
Exactly. And that can contribute to the thickening of the basement membranes in blood vessel.
Right, and we know that's a key feature of DR.
Yeah, and it can also make the blood vessels less responsive to nitric oxide.
Oh, which is important for vasodilation.
Right, so it makes it harder for the blood vessels to relax and dilate properly.
Okay, so that's pretty significant.
Yeah, and here's another interesting thing about this hexosamine pathway.
Okay.
This UDP-GLC-NEC and OGLC-acylation, they can actually affect RNA polymerase II.
RNA polymerase II, that's the enzyme that makes messenger RNA, right?
Exactly. So it's involved in the very first step of gene expression. And when RNA polymerase II gets OGLC-acylated, it can change how efficiently it transcribes genes.
So it's like messing with the master control switch for gene expression.
Exactly. And that can have all sorts of downstream effects on the cell.
Wow, that's pretty powerful.
Yeah, so this hexosamine pathway, even though it's not always talked about, it's really a major player in how high glucose disrupts cellular function.
Okay, so that's 4 pathways now. The polyol pathway, AGEs, PKC activation,
Yeah.
and the hexosamine pathway.
Right.
And they all seem to be contributing to oxidative stress and inflammation in the retina.
Exactly.
Okay, so are we missing any key players in this complex story of DR development?
Well, there's one more that we need to talk about, which is ADP ribose
Okay.
polymerase, or PRP.
PRP.
It's kind of like a cellular damage control system.
Okay,
So when there's DNA damage, PRP gets activated and tries to repair it.
that sounds good.
Yeah, but the problem is, in the context of diabetes, there's just too much DNA damage happening because of the oxidative stress. So PRP gets overactivated and starts using up a lot of NAD plus A.
NAD plus A, that's another coenzyme, right?
Yeah, it's really important for a lot of different cellular processes.
Okay.
And when PRP depletes NAD plus A, it can actually worsen the damage.
Oh, wow. So it's like the damage control system backfires.
Exactly.
Okay, and on top of that, PRP activation also inhibits an enzyme called GAPDH,
Right.
which is involved in glucose metabolism, right?
Yeah, it's a key enzyme in glycolysis.
Okay.
So inhibiting GAPDH further disrupts the cell's energy production.
So it's like a vicious cycle.
Yeah, and all of this contributes to endothelial cell dysfunction, which obviously isn't good for the blood vessels in the retina.
Okay, so PRP activation is another important piece of the puzzle.
Right.
And all these pathways we've talked about, the polyol pathway, AGEs, PKC activation, the hexacenine pathway, and now PRP, they all converge on these two key processes, oxidative stress and inflammation.
Exactly.
And those are the ultimate drivers of the vascular damage we see in Dakar.
Right. And, we've talked a lot about hyperglycemia being a major trigger for all of this,
Right.
but it's not the only source of oxidative stress in Dogger.
Oh, really?
Yeah, so remember how hyperglycemia activates PKC, which then activates NADPH oxidase? Well, NADPH oxidase is a major source of reactive oxygen species, or ROS.
ROS, those are the things that cause oxidative damage, right?
Exactly. So it's like a self-perpetuating cycle.
Yeah.
Hyperglycemia leads to more ROS production, which then causes more damage and further contributes to the development of Dogger.
So it's like a runaway train.
Yeah, pretty much.
Okay, so we've got oxidative stress, but then there's also inflammation. How does that play into all of this?
Well, inflammation is a really complex process, but basically it involves the recruitment of immune cells to the site of injury.
Okay.
And those immune cells release all sorts of inflammatory mediators, cytokines, chemokines, all sorts of things.
Yeah.
And in Dogger, this inflammatory response actually ends up doing more harm than good.
Oh, how so?
Well, it contributes to the breakdown of the blood-retinal barrier, leading to leakage of fluid and protein into the retina.
Okay, that explains the macular edema.
Right.
Okay.
But it also promotes the growth of those abnormal blood vessels.
Oh, so it's fueling neovascularization too.
Exactly.
Okay.
And inflammation can actually damage the retinal neurons directly.
So it's not just affecting the blood vessels.
No, it's damaging the retina on multiple levels.
Okay.
And what's really interesting is that inflammation and oxidative stress are closely linked. Like they kind of feed off each other.
Okay, how so?
Well, oxidative stress can actually trigger inflammation. And then the inflammatory cells that get recruited to the retina can produce more ROS, which further increases oxidative stress.
So it's like a vicious cycle of damage.
Exactly. It's a really complex interplay of different pathways and processes.
And it all ultimately leads to those hallmark vascular abnormalities we see in DR.
Right.
Like the microvascular occlusion, the ischemia.
And then the neovascularization.
And the formation of IRMAs.
Exactly.
Those intraretinal microvascular abnormalities. Right. Okay, so we've covered a lot of ground on the pathophysiology of DR.
Yeah, it's complex stuff.
But it's really important for understanding how the disease develops and why certain treatments work the way they do.
Absolutely.
Now, let's shift gears and talk about screening.
Sure.
What are the latest recommendations for DR screening, and how do we make sure we're catching this early in our patients?
Well, the most recent guidelines we have to go by are the 2018 ICOATA recommendations for DR screening.
Okay.
And really, the core message there is that for adequate screening, you need both a visual acuity assessment and a thorough retinal exam.
Okay, so that's the standard we should be aiming for.
Yeah, definitely.
Now, in terms of visual acuity, the guidelines mention a few different acceptable methods, right?
Right. So they say you can either do a refracted visual acuity measurement using a standard eye chart at the typical distance, or you can do a presenting visual acuity measurement with either a near or a distance chart.
Okay.
And if the patient's acuity is reduced, you can use a pinhole to try to improve it.
Right. Makes sense.
Or you can use a handheld chart that's equivalent to 612, which is a pretty common approach, especially in busy clinics.
Yeah, I think a lot of us use those handheld charts.
Yeah, they're pretty handy.
So it's good to know that those are all acceptable methods according to the guidelines.
Yeah, it gives you some flexibility.
Now, what about the overall screening rates here in the U.S.? Are we doing a good job of catching DR early in our diabetic population?
Well, the latest data we have shows that only about 62.3% of adults with diabetes are actually getting screened for DR annually.
Oh, wow, that's not great,
Yeah, it's a bit concerning.
especially considering how common diabetes is and how serious DR can be.
Exactly. And what's even more worrying is that there are these really significant disparities in screening rates.
Oh, what do you mean?
Well, certain groups are less likely to get screened than others.
Okay, like who?
So people with lower levels of education, lower income, minorities, people living in rural areas, and people without health insurance, they're all less likely to get screened.
Wow, so it's really hitting those vulnerable populations the hardest.
Yeah, it's a real equity issue.
And you said health insurance status has the biggest impact?
Yeah, it seems to be the most significant factor.
That's really concerning.
Yeah, it highlights the need for more proactive outreach and better access to care for those who need it most.
Definitely. So we've got a lot of work to do to improve those screening rates and make sure everyone's getting the eye care they need.
Absolutely.
Now, are there any national efforts or goals aimed at addressing this issue?
Yeah, actually, the Healthy People 2030 initiative has set some specific targets for DR screening.
Oh,
Their goal is to increase the proportion of adults with diabetes who are getting screened annually from that 62.3% we talked about up to 67.7%.
Okay.
And they also want to cut the rate of vision loss due to DR in half.
Wow, that's ambitious.
Yeah, it's a big goal, but it's certainly achievable if we all work together.
Definitely. Now, in terms of the initial retinal exam, when should we be seeing these patients for the first time?
Well, the AAO's preferred practice pattern recommends that for patients with type 1 diabetes, we should see them for their first eye exam 3 to 5 years after they're diagnosed.
Okay.
And for people with type 2 diabetes, it's recommended at the time of diagnosis.
Okay, that makes sense.
And for pregnant women who already have type 1 or type 2 diabetes, we should see them soon after they get pregnant.
Ideally in the first trimester.
Right, exactly.
But for women who develop gestational diabetes without any pre-existing diabetes, they don't necessarily need a routine retinal exam.
That's right.
Okay, that's good to know.
It's always helpful to clarify that for outpatients.
Definitely. Now, once we've done that initial screening, what are the guidelines for referring patients to a retina specialist?
Okay, so in places where we have plenty of resources, the 2018 ICOATA guidelines give us pretty specific criteria. So if a patient has no DR or only mild NPDR without macular edema, we don't necessarily need to refer them right away.
Okay.
They can usually just be followed up by their primary care physician or general ophthalmologist.
Right.
But for moderate NPDR, we do want to refer them to a retina specialist, and we recommend following up in about 3 to 6 months.
Okay.
And for severe NPDR or PDR, obviously we want to refer them urgently.
Right.
Ideally within 3 months for severe NPDR and within 1 month for PDR.
Okay, so those are the time frames we should be aiming for.
Yeah, and of course, if there's any DME, whether it involves the center of the macula or not, we want to refer them to a retina specialist as well.
Right, and OCT is crucial for managing DME, right?
Oh, yeah, absolutely.
Yeah.
It helps us assess the severity of the edema, track its response to treatment, and guide our follow-up intervals.
Okay.
So it's definitely a must-have tool for any retina specialist.
Now, what about in resource-limited settings? Do these referral guidelines change at all?
Well, the ICOATA guidelines acknowledge that we might need to be a little more flexible in those situations.
Okay.
So for patients with no DR or mild to moderate NPDR who are considered low risk, we might be able to extend the screening intervals to 1 to 2 years.
Okay, so stretching things out a bit when resources are tight.
Yeah, exactly. But for severe NPDR or PDR, those patients still need to be referred urgently, even in resource-limited settings.
Okay, so those are the ones we can't compromise on.
Right.
Now let's talk about some of the newer technologies that are changing the way we screen for DACAR.
Sure.
Retinal photography is becoming increasingly common.
Yeah, it's definitely gaining popularity.
And it seems like ultra-wide field photography is generating a lot of buzz in the field.
Oh yeah, UWF photography is really exciting.
What's so special about it?
Well, for one, it gives us a much wider view of the retina than traditional photography.
Okay.
So we can see more of the periphery, which is where a lot of the early DR changes can happen. And studies have shown that UWF photography is just as good as traditional methods at detecting DR.
Okay, so it's not sacrificing accuracy for that wider field of view.
Exactly.
That's great.
And another advantage is that it takes less time to capture the images.
Oh, that's a big plus in a busy clinic.
Yeah, definitely.
So it sounds like UWF photography has a lot to offer.
Yeah, it's definitely a tool that's worth considering.
Now, what about artificial intelligence? Seems like everyone's talking about AI these days,
Oh, yeah, AI is everywhere.
and it's starting to make its way into DR screening as well.
Right, we're actually seeing tangible applications of AI in our practices now.
Oh, really?
Yeah, there are already a few FDA-cleared AI systems out there that can analyze retinal images and tell us if a patient needs to be referred to an ophthalmologist.
Wow, that's pretty amazing.
Yeah, it's pretty incredible.
So how do these systems work?
Well, they're basically trained on thousands of retinal images that have been graded by human experts, and they learn to recognize the patterns that are associated with DR.
Uh-huh.
So when you feed a new image into the system, it can analyze it and give you a prediction of whether or not the patient has more than mild DR.
Wow, so it's like having a virtual expert on hand 24/7?
Yeah, kind of.
That's incredible.
But it's important to remember that these systems do have some limitations.
Okay, like what?
Well, they're not as good at detecting DR in pregnant women
Uh-huh.
or in patients who already have severe DR.
Okay, so there are some specific populations where they might not be as accurate,
Right.
but overall, how do they compare to human graders?
Well, studies have shown that they're actually pretty comparable in terms of their ability to detect referable DR.
Okay, so they're getting pretty good.
Yeah, they're definitely getting there.
That's really exciting.
Yeah, and the technology is only going to get better from here.
Now, are these systems compatible with UWF images?
Well, most of the currently available systems aren't designed for UWF images, but there's a lot of research going on in that area, so I think we'll start to see UWF compatible AI systems in the near future.
That'd be great.
Yeah, it would really expand the potential of AI in DR screening.
Now, what about the possibility of home-based screening using AI?
Oh, yeah, that's a really interesting area.
It seems like that could be a game changer for reaching people who don't have easy access to eye care,
Absolutely.
like people in rural areas or underserved communities.
Right.
But I imagine there are some challenges with that as well.
Yeah, definitely. One of the biggest challenges is patient acceptance.
Uh-huh.
Are people going to be comfortable using these devices at home?
Right, and trusting the results.
Exactly.
And what about data privacy?
Oh, yeah, that's a huge concern.
Making sure that sensitive medical information is protected.
Absolutely.
And then there are the medico-legal implications of AI-driven diagnoses.
Right, that's a whole other can of worms.
So it sounds like there's a lot to work out before home-based AI screening becomes a reality.
Yeah, but I think it has huge potential.
Definitely.
It's definitely something to keep an eye on.
Now, one last thing on screening. I've heard about using AI for predictive modeling.
Oh, yeah, that's another really exciting area.
But what is that exactly?
Basically, it's using AI to predict which patients are at highest risk of developing DR or having their DR progress.
Okay.
So we can tailor their screening intervals accordingly.
Oh, that makes sense. So instead of everyone getting screened annually, we could focus our resources on the patients who need it most.
Exactly.
That's a great idea.
Yeah, it has the potential to make screening much more efficient and effective.
Okay, so it sounds like the future of DR screening is going to be a really interesting mix of traditional methods and these cutting-edge AI technologies.
Yeah, I think it's going to be a really exciting time in our field.
Okay, so we've talked about how to detect AR.
Right.
Now let's talk about how to manage it.
Okay.
So what are the cornerstones of treating diabetic eye disease?
Well, the most important thing, the absolute foundation of everything we do, is making sure our patients are managing their systemic conditions.
Right, their diabetes, their blood pressure, their cholesterol.
Exactly. So we need to be working closely with their primary care doctors, their endocrinologists, to make sure they're getting the best possible systemic care.
Definitely.
And we know from those landmark studies like the DCCT and the UKPDS that good glycemic control can make a huge difference in the development and progression of DOCR.
Right, the DCCT showed that intensive glycemic control could actually reduce the risk of developing retinopathy by over 70%.
Exactly. Those were really groundbreaking studies.
And they also showed that even if retinopathy does develop, good blood sugar control can slow down its progression.
Absolutely. And now we have this concept of metabolic memory.
Metabolic memory.
Yeah, it's basically the idea that the early years of diabetes have a really big impact on the long-term health of the blood vessels.
Oh, interesting.
So even if someone gets their blood sugar under control later on, the damage that was done early on can still have lasting effects.
So it's like the body remembers those periods of high glucose.
Exactly.
Okay, so that really underscores the importance of tight blood sugar control from the very beginning.
Absolutely. We can't stress that enough.
Now, in terms of specific targets, what's the general recommendation for HbA1c?
Well, the American Diabetes Association recommends aiming for an HbA1c of less than 7%.
Okay.
But it's important to remember that this is just a general guideline.
Uh-huh.
We need to individualize our targets for each patient.
Right, taking into account their overall health, their age, their other medical conditions.
Exactly.
And there was that study, the Accord study, that raised some concerns about overly aggressive blood sugar lowering, right?
Yeah, that study showed that in some patients, trying to push their HbA1c too low could actually increase their risk of death.
Wow, so it's a delicate balance.
Definitely. We need to be careful not to overcorrect.
Okay, so systemic control is paramount. But how do we actually classify DR to guide our specific treatment strategies?
Well, as we talked about earlier, the main categories are NPDR and PDR. And
Yes.
within NPDR, we further categorize it as mild, moderate, or severe.